Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Active immunity explained: types, mechanisms, and comparison with passive immunity. Table of contents. Introduction of active immunity. Characteristics of active immunity. Types of active immunity. Mediators of active immunity. Active immunity versus passive immunity. Introduction of active immunity. Active immunity develops when the body produces its own antibodies in response to exposure to a pathogen or foreign antigen. It provides long-term protection and resistance against infections. Active immunity is induced when the immune system responds to a foreign antigen by producing antibodies. The immune system recognizes a pathogen, activates immune cells, and generates a defense mechanism against it. It can be acquired through clinical or subclinical infection, which refers to previous exposure to an antigen that may or may not have caused noticeable symptoms. It can also be induced through immunization by vaccination with leave or killed infectious agents or their antigens. Exposure to microbial products such as toxins and toxoids can also trigger an immune response. The immune system stimulates an immune response against foreign antigens by activating B cells, which produce antibodies, and T cells, T helper and cytotoxic T cells, which help eliminate infected cells. There's an initial lag period before immunity is fully developed, but once established, active immunity provides long-lasting protection. The primary immune response may be slow, but memory cells ensure a faster and stronger response against future exposures. Active immunity plays a crucial role in protecting individuals from infections and is the foundation of vaccination programs worldwide. Characteristics of active immunity. Active immunity requires exposure to a pathogen or its antigen. This exposure stimulates the production of antibodies, which mark infected cells for destruction by lymphocytes. Cells involved in active immunity include T cells, cytotoxic T cells, helper T cells, memory T cells, and suppressor T cells, B cells, memory B cells and plasma cells, and antigen-presenting cells, B cells, dendritic cells, and macrophages. There's a delay between exposure to the antigen and the development of immunity. The first exposure triggers a primary immune response, which takes time to develop. Upon subsequent exposure, the immune system mounts a secondary response, which is much faster and stronger. Active immunity provides long-term protection, often lasting a lifetime. Some potential side effects of active immunity include autoimmune diseases and allergic reactions in certain cases. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Types of active immunity. A. Natural active immunity. Natural active immunity is acquired when an individual is exposed to a pathogen, either clinically with symptoms or subclinically without noticeable symptoms. Exposure to a live pathogen leads to infection, triggering the immune system to develop a long-term immune response. When a microbe bypasses the body's primary defenses, such as the skin or mucous membranes, it activates B cells, which produce antibodies to fight the infection. The adaptive immune response takes days or weeks to develop, but can provide long-lasting or lifelong immunity. For example, individuals infected with the hepatitis A virus, HAV, and subsequently recovering develop a natural active immune response, often leading to lifelong immunity. B. Artificial active immunity. Artificial active immunity is induced through vaccination, where an antigen is introduced into the body to stimulate an immune response. The administration of vaccines, such as the hepatitis A vaccine, triggers an immune response that provides long-term or lifelong protection against the disease. Vaccination is a deliberate process that introduces a harmless form of an antigen derived from a pathogen to activate the immune system without causing disease. Immunizations are highly effective because they leverage the immune system's natural specificity and ability to form memory cells. The principle behind vaccination is to stimulate protective immunity while avoiding the harmful effects of the actual pathogen. Artificial active immunity is achieved through various vaccines, which may be live vaccines, killed vaccines, or vaccines containing bacterial products. Mediators of active immunity. Active immunity is mediated by humoral immunity and cell-mediated immunity, both of which play crucial roles in eliminating pathogens. Humoral immunity, mediated by antibodies found in blood and mucosal secretions. B-cell plasma cells secrete antibodies that recognize microbial antigens, neutralize their infectivity, and mark them for elimination. This type of immunity is the primary defense against extracellular pathogens. Cell-mediated immunity, mediated by T helper cells and cytotoxic T lymphocytes. T helper cells secrete cytokines that activate phagocytic cells, enhancing their ability to engulf and destroy pathogens. Cytotoxic T lymphocytes play a key role in killing virus-infected cells and tumor cells by targeting altered self cells. This immune response is particularly important against intracellular bacterial and protozoal infections. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Active versus passive immunity. Active immunity is developed by the host's immune system in response to exposure to an antigen. In this process, antibodies are produced as a reaction to infection or immunization. It involves both humoral and cell-mediated immune responses. Active immunity can be acquired naturally through infection or artificially through vaccination. However, it takes time to develop due to the immune response lag period. Once established, active immunity provides long-lasting and effective protection, as it involves immunological memory. Booster doses can further enhance this immunity. In some cases, active immunity may lead to temporary suppression of the immune response, known as the negative phase. However, it is ineffective in immunodeficient individuals. Active immunity is primarily used for preventive purposes, but may contribute to autoimmune disorders and allergic reactions in rare cases. On the other hand, passive immunity is acquired without active involvement of the host's immune system. Instead, preformed antibodies are transferred directly into the body. This type of immunity does not involve cellular responses, but is based entirely on ready-made antibodies. Passive immunity can be natural, such as the transfer of maternal antibodies through the placenta, or artificial, such as the administration of immunoglobulins. Unlike active immunity, passive immunity provides immediate protection without a lag period. However, it is temporary, as the antibodies degrade over time, and no immunological memory is formed. Subsequent doses of passive immunity treatments have reduced effectiveness due to antibody clearance. Unlike active immunity, passive immunity does not cause a negative phase and can be used for immunodeficient individuals. It is mainly employed as a therapeutic measure after exposure to a pathogen. However, it may lead to side effects, such as serum sickness, in certain cases. Both active and passive immunity play crucial roles in protecting the body from infections, but their mechanisms, duration, and applications differ significantly. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!